This video is part 2 of the lecture on the basic model of multiple linear regression. Let's look at another example, which shows that multiple linear regression can be used to better understand relationships among variables. In the previous session, we investigated a hypothesis on age variation in systolic blood pressure. In this example, we start with a hypothesis about the relationship between blood sugar and blood pressure. Suppose that, for some reason, we think that a high level of blood sugar should raise the level of blood pressure. If we have cross-sectional data that includes systolic blood pressure and serum glucose, we will probably start with plotting blood pressure against serum glucose, and then we may do linear regression of blood pressure on serum glucose. The regression model should look like this. Here, Y is systolic blood pressure, and X2 is serum glucose. A and B2 are parameters to be estimated from data. This is the result of the regression analysis using the NHANES data on persons aged 20 to 64. According to this regression model, systolic blood pressure is predicted as 80.55 plus 0.37 times X2, which is serum glucose. The value of the regression coefficient, 0.37, means that in the data, on average, or typically, an increase of systolic blood pressure by 0.37 mm of mercury corresponds to a unit increase of serum glucose, i.e., an increase of serum glucose by 1 mg per deciliter. This indicates a positive correlation between systolic blood pressure and serum glucose. But we saw, in session 1, that systolic blood pressure is strongly associated with age. And serum glucose may be related to age as well. So, although the data show a positive correlation between systolic blood pressure and serum glucose, the correlation may be partly due to the combination of their relations to age. Then, the next question is, how is systolic blood pressure related to serum glucose among people at the same age? There are several ways to estimate the relation between systolic blood pressure and serum glucose among people at the same age. First, we can do regression of systolic blood pressure on serum glucose for those aged 25, for example, and the same regression analysis again for those aged 26, and then another for those aged 27, and so on. But in this way, we will have to do many regression analyses. So we need a huge sample. Also, we get a regression result for those aged 25, another result for those aged 26, another for those aged 27, and so on. So we will end up with many estimates of the regression coefficient, which need to be summarized using a proper statistical procedure. Second, we can adopt wider age intervals such as 10-year age groups, and do the regression analysis for those in the 20s, another regression for those in the 30s, and another for those in the 40s, and so on. This requires a smaller sample, but we still need a fairly large sample, because we need to do regression for each of several 10-year age groups. Also, we are controlling for age in a more crude way than before, because in this approach, age variations within each 10-year age group are ignored. But age differences within each 10-year age group may have some effect. For example, those aged 59 may tend to have higher levels of systolic blood pressure and serum glucose than those aged 50. In addition, we still have to statistically summarize different estimates of the regression coefficient for the 10-year age groups. Thirdly, we can do multiple linear regression of systolic blood pressure on serum glucose and age. The regression model has a parameter, the partial regression coefficient for serum glucose, which summarizes age-adjusted relation between serum glucose and systolic blood pressure. This is a practical and convenient way, which does not require a very large sample, and produces just one parameter estimate summarizing age-controlled relation between serum glucose and systolic blood pressure. This slide shows two regression models of systolic blood pressure. Model 1 is regression of systolic blood pressure on serum glucose only, and Model 2 is regression of systolic blood pressure on age and serum glucose. In Model 2, a unit difference in serum glucose is associated with a difference in systolic blood pressure by B2, holding age constant. This phrase, holding age constant, can be interpreted as follows. According to the Model 2 equation, when the value of X2 increases by 1, then Y increases by B2. But it is so, as far as X1 remains unchanged. Suppose that the value of X1 changes when the value of X2 changes. Then, when the value of X2 increases by 1, the corresponding change in Y may not be B2. Again, if X2 increases by 1, Y increases by B1, as far as X1 remains constant. What about the last term in Model 2, E? It is assumed to represent some random variations, the mean value of which is 0. So when x2 increases by 1, the corresponding increase in y may be greater than b2, if e is positive, or smaller than b2, if e is negative. But on average, the increase in y should be b2, according to Model 2. We study about e more in Session 5. This is an intuitive explanation. There is a more mathematical explanation, which we will study in a later session. This slide shows results of 3 regression analyses of systolic blood pressure. The first is on age only, the second is on serum glucose only, and the third is on age and serum glucose together. Note that the regression coefficient for serum glucose became smaller from 0.37 to 0.31 by incorporating age into the model. Why did the coefficient for serum glucose decrease from 0.37 to 0.31 when age was included in the regression equation? In order to explore this question, I did 3 more simple regression analyses of systolic blood pressure on serum glucose. The estimated regression coefficient was 0.37 for the broad age range from 20 to 64. I split the age range into 3 narrower age ranges of 20 to 34, 34 to 49, and 49 to 64, and obtained an estimate of the coefficient for each of the ranges. We may expect that 0.37 should be like an average of the estimated coefficients for the 3 narrower age groups. But actually, as shown here, all of the 3 estimated coefficients were lower than 0.37. What was happening? Let's explore, using simulated data. This shows systolic blood pressure by serum glucose in the simulated data. For visual clarity, their correlation was made stronger than it actually was. The red line is the regression line fitted to all points in the graph. Now let's introduce age into the graph. The black points indicate respondents who are in the 20s, the red points indicate those in the 30s, the green points in the 40s, the blue points in the 50s, and the light blue points in the 60s. We see that the 5 clusters of points are at different locations in this graph. This figure shows systolic blood pressure and serum glucose are associated with age. Systolic blood pressure is associated with age. For example, the cluster of the light blue points are at a higher position than the cluster of blue points, indicating that those in the 60s tend to have higher blood pressure than those in the 50s. The cluster of the green points are at a higher position than the cluster of the red points, indicating those in the 40s tend to have higher blood pressure than those in the 30s. Serum glucose is associated with age as well. The cluster of the light blue points are on the right of the cluster of blue points, indicating those in the 60s tend to have higher levels of serum glucose than those in the 50s. The cluster of the red points are on the right of the cluster of the black points, indicating those in the 30s tend to have higher levels of serum glucose than those in the 20s. Here, a straight line is fitted to points of each color, indicating that systolic blood pressure and serum glucose are positively correlated in each of the 5 age groups. But the slopes of these lines are flatter than the slope of the straight line fitted to all of the points, which we saw 2 slides before. This suggests that when the relation between systolic blood pressure and serum glucose is adjusted for age, their relation is not so strong as their relation not adjusted for age. The simulated example in this schematic presentation help us to understand why the value of regression coefficient for serum glucose became smaller when age was included in the regression model. The black line represents the bivariate relation between systolic blood pressure and serum glucose. On average, an increase in systolic blood pressure by 0.37 corresponds to a unit increase in serum glucose. But this relation is partly due to age, because systolic blood pressure is positively related to age, and serum glucose is positively related to age as well. So, the relation between systolic blood pressure and serum glucose can be split into 2 kinds of relationships: their relation with each other among people at the same age, which may be called their age-adjusted or age-controlled relation, and their relation with each other, resulting from the fact that both of them are related to age. These 2 relationships are represented by the green line and the purple lines, respectively. As shown in a previous slide, the regression analysis of systolic blood pressure on serum glucose and age indicated that, among the people at the same age, on average, an increase in systolic blood pressure by 0.31 corresponds to a unit increase in serum glucose. A special statistical method called path analysis shows that through age, an increase in systolic blood pressure by 0.054 corresponds to a unit increase in serum glucose. Therefore, the simple regression coefficient for serum glucose, 0.37, is decomposed as 0.31, representing the age-adjusted association between systolic blood pressure and serum glucose, plus 0.054, representing their association through age. So this example illustrates how linear regression is used for understanding relationships among three or more variables. A few words on the terminology and notations may be of help. This slide shows a model of linear regression with two independent variables, x1 and x2. The upper equation is for the predicted value of the dependent variable, y hat, and the lower equation is for the observed value of the dependent variable, y. A is the estimated mean value of y, when both x1 and x2 are 0. B1 is the expected difference in y, associated with a unit difference in x1, when x2 is fixed. B2 is the expected difference in y, associated with a unit difference in x2 when x1 is fixed. By the way, you may have noticed that so far, in some slides, alpha, beta, and epsilon were used, and in other slides, a, b, and e were used. Actually, the distinction had some meanings, but the distinction is not important in this session. It will become more relevant in sessions 4 and 5. Different papers and textbooks use different names for B in the linear regression model, such as partial regression coefficient, beta coefficient, slope coefficient, regression weight, and so on. Basically, these have the same meaning. Also, different phrases are used when the partial regression coefficient is interpreted. Here, B1 summarizes the relation between y and x1, controlling for x2, or adjusted for x2, or holding x2 constant. These phrases have essentially the same meaning. So far, we have been studying linear regression with two independent variables, x1 and x2 here. The model can be easily extended to three or more independent variables, by adding b3 times x3, b4 times x4, and so on. Why is linear regression called linear? In this slide, the third equation is an expression of y hat, in linear regression with three or more independent variables. In mathematics, this form means that y hat is a linear combination of x1, x2, and the other x's. Note that the equation has no multiplicative term such as x1 times x2, no power such as x1 squared, or no mathematical conversion of x's such as log x1 and tangent x2. It is a linear equation. We are searching for a best-fitting straight line for our data. In the next lecture, we will talk about controlling for confounding factors.